So hello all. Yes, in the previous class we had discussed about the structure of nitrogenous bases, that how these purines and pyrimidine rings are formed usually. So today we are going to discuss about how the nucleotide structures are formed, how nucleoside structures are formed, and we are going to discuss in details what are the bonds involved in forming these structures. All right. So today on the board I have made a nucleotide structure, and I'll also make it for you once again so it will be more clear for you people. Okay, so this is the structure of adenine as we had drawn earlier, that adenine is a double ring structure. It is a purine ring, right? So it's a double ring structure in which it is called, adenine is called what? Adenine is called 6 amino purine. So at the sixth position of the purine ring, the amino group is being attached. So it is called 6-aminopurine. Now we have to draw the structure of nucleotide. So what is a nucleotide? Nucleotide is sugar plus base plus phosphate group. Okay. So when we talk about RNA and DNA, so there are differentiating features between DNA and RNA. Very first basic difference is that DNA is double stranded, RNA is single stranded. That we all know, right? RNA although can form secondary structures, but it is single stranded. Okay. Then we have next very important feature, that in the sugar, in case of the second position, for example, this is the ribose sugar, this is the deoxyribose sugar. Actually, I'll tell you what is deoxy and what is ribose. This on the second prime position. So why do we write prime? So that we cannot get confused between the numbering of the base and the sugar. So the base numbering is 1, 2, 3, 4. Then the sugar numbering is 1 prime, 2 prime, 3 prime, so that we can differentiate between the numbering of the base and the sugar. Okay. So the sugar numbering is like this: 1 prime, 2 prime, 3 prime, 4 prime and 5 prime. Alright, so we have, like, at the 2 prime position of the sugar, we have deoxyribose. That means there is lack of oxygen atom in the second position of the sugar, of the ribose sugar. That's why DNA is called deoxyribonucleic acid. The name itself suggests कि structure में at the 2 prime position of the sugar there is deoxy group, not the oxy group. All right, so we have deoxyribonucleic acid. But in the case of RNA, we have ribonucleic acid. We do not have the deoxyribonucleic acid. Now the third most important differentiating factor is that DNA has thymine instead of uracil. In the RNA we have uracil. Now there is a very important reason why DNA has like DNA has thymine and not uracil. We'll discuss it further when we'll discuss the structure of RNA and DNA in detail. Firstly, we have to learn the structure of nucleotides. So what are nucleotides? They have sugar, base and phosphate group. Okay. Bases we had discussed in the previous class. Now I'm going to tell you the structure of the sugar. So the structure of the sugar that is present in your nucleic acid is your ribose sugar. Which sugar is present? Ribose sugar is present. So when we talk about the ribose sugar, it is actually forming this kind of structure. It is forming this kind of structure. Pentose ring is there, and we have OH, H, we have H, OH, H, OH, then we have CH2OH and H. Okay, so we have OH, H, we have H, OH. So on the 2 prime position, there is a differentiating factor that for RNA, we have oxygen atom, but for the DNA, we don't have the oxygen atom. We have only the H atom for the 2 prime position. Now we are going to learn about the third group that is present in the nucleotide, that is the phosphoric acid or the phosphate group. So phosphate group is also present in your structure. Okay, now we are going to learn what kind of bonds are involved in the structure of DNA, okay, in forming the nucleotide. As I told you that the difference comes only on the basis of the nitrogenous bases that we are adding. That is, it can be purine or pyrimidine, it can be AG or CUT, but the sugar phosphate backbone remains the same. Now how this sugar phosphate backbone is linked to the bases and how sugar is linked to the phosphate group, we need to learn that now. Okay, So first of all, we will see how bases are linked to the sugar. Okay. So when we talk about the linkage between the sugar and the bases, the bond that is formed is known as the glycosidic linkage. Now, what is this glycosidic linkage? Glycosidic linkage is that linkage which is formed when what happens? When there is removal of water. When the bond is formed when there and there is resulting what? Resultant, there is a removal of water. Okay, so when we talk about this structure, right, there was OH here and it was NH here. So what will happen? The water will remove and this bond will form. This glycosidic linkage will be forming. Glycosidic linkage. Let me just rub whatever is written here because now I have already explained it to you. Glycosidic linkage. Which linkage is involved? Glycosidic linkage. And it is formed by what? It is formed by removal of water. So water remove ho jayega and then there will be a linkage that is formed which is known as glycosidic linkage. Now you have to mark this very important point that in the case of purine, it is joined to the sugar by N9 position. By which position? N9 position. But in the case of pyrimidines, it is joined by N1 position. So this glycosidic bond is formed between the sugar and base for purines. Very important to note this thing because when you draw the structure, it will be very crucial ki kahan se you have to join the sugar to the base. All right. So for example, I give you the question: draw the structure of 7-methyl guanosine. It is a nucleoside, right? You have to draw the sugar and the base, right? So how will you draw that? You have to know that how to join the sugar to the base. To what position you need to join the sugar to the base. Now guanosine is a purine. Now how will you join the purine? By the N9 position of the ring. So N9 position is joined to this first position of the deoxyribose sugar or ribose sugar. When in the case of RNA, we say ribose sugar. In the case of DNA, we say deoxyribose sugar. In the question, if it is not mentioned anything, it is mentioned 7-methyl guanosine. So you have to write for the RNA. You don't have to remove the oxygen atom from the ring. Okay. By default, you will put the ribose sugar only. Okay. Now N9 position for the purines and for the pyrimidines, it is N1 position for the ring. जो N1 position होगी ring की, the N1 position of the ring for the pyrimidine will be joined to the first carbon of your ribose sugar or the deoxyribose sugar. Now most important bond that is formed by your polymerases also during replication, as you know that DNA is replicated for it to be transferred into the daughter cells. In the process of cell cycle, what happens? There is a cell, it divides into two daughter cells. So if it has to be fair division, then what has to happen? The genome has to be divided faithfully. So in that process, the DNA replication takes place. It is a very faithful process in which whole genome is replicated into two copies. One goes to one daughter, other goes to other daughter cell. Okay. Now in the DNA replication, the most crucial enzyme that we have is your DNA polymerases. Now what are these DNA polymerases? These DNA polymerases are actually forming the phosphoester linkage. Now you need to learn what is the phosphoester linkage. Now what are we going to do is, what are we going to do? Yeah, we're going to learn how is this phosphoester linkage being formed. First of all, there is a phosphoester linkage being formed at 3' position. Always remember that the genome, in the genome, when the DNA replication occurs, the polymerase always adds group to this 3' position. This is the structure of the nucleotide. This is the structure of the 3' position of the sugar. So this is the first nucleotide being added. For example, this is one strand of the DNA. तो एक स्ट्रैंड पे एक बेस अटैच हो चुका है, राइट? नाउ द सेकंड बेस विल कम, ऐसे ही फॉस्फेट ग्रुप आएगा एंड इट विल अटैच टू द 3' पोजीशन ऑफ योर स्ट्रैंड. तो एक स्ट्रैंड में, डॉटर स्ट्रैंड में क्या होगा? The strand that is opposite to your template strand, 3' position पे, the next base will be attached and that will be the phosphoester linkage that will be formed by your DNA polymerases. Now there is a bond which is formed here also, that is between the alcohol and the acid. Now the alcohol and the acid, because this is CH2OH, as I had shown in the structure of purine, here it is CH2OH and here it is the phosphoric acid group. Now the phosphoric acid group and the CH2OH, when they will form, they will form the bond, it will be actually the for ester bond. Why we call the ester bond? Because it is the bond which is formed between the alcohol and the acid moiety. So when the alcohol and the acid join, they form what? Esters. So it is the ester bond which is formed between the phosphate and the sugar residue. Okay, so this is known as the phosphoester bond. Now here also the bond will form between the phosphate and the sugar residue, so that will also be called the phosphoester bond. That's why we say that it is called phosphoester bond. Okay, so this is what, how our nucleotide structure is formed. We have the base, we have the sugar, we have the phosphate group. This is joined by the glycosidic linkage, this is joined by the ester linkage. So we are done with the structure of nucleotides. Now further we will do the structure in the nucleosides. Like, what are nucleosides? Now nothing is there in the nucleosides, only the sugar and the base is attached. When you remove this phosphate group, only sugar and the base is there. That is known as your nucleoside structure. All right, so we are now going to learn some examples how to draw the structure of nucleoside base. Now we're going to discuss one example of 7-methyl guanosine. So it is a nucleoside because we are not mentioning the phosphate group. I have not said that you have to draw the structure of guanylate. I have said 7-methyl guanosine. There is a difference between guanine, guanosine and guanylate. When we talk about guanine, then it is the nitrogenous base. When I say guanosine, it is your nucleoside. When I say guanylate, then it is your nucleotide. Now we're going to discuss about 7-methyl guanosine structure. Now what is with the 7-methyl guanosine structure? We have the guanosine group. As I told you, in the guanosine, in the structure of adenine, it was 6-aminopurine. You have to shift the amino group niche. So when the amino group is shifted niche, the oxygen atom comes here and we have to draw 7-methyl guanosine. It is a very like a like a modification that happens on the 5 prime end of the mRNA. During the transcription, what happens, there is this modification at the 5 prime end of the mRNA, that is methylated guanines are added on the 5 prime end of the mRNA. Okay, so it is that kind of modification only. So in the structure of 7-methyl guanosine, on the 7th position of the guanine nitrogenous base, you have to add this methyl group. So when we add the methyl group to the 7th position of this base, then what it will form? 7-methyl guanine. Okay. Now we have to draw the structure of guanosine. I told you that guanine is a base, guanosine is a nucleoside. Nucleoside is sugar plus base. So sugar plus base mein we have the sugar, and I have not mentioned that you have to write deoxy or ribose. So for the default, you have to write the ribose sugar only. So we have to mention the ribose sugar only. We will not mention the deoxyribose sugar. Okay. So we have the 7-methyl guanine base and we have the ribose sugar. So when they both are attached by the glycosidic bond, and the glycosidic bond is formed between N9 position and the first position of your sugar. N9 of the base and 1 prime of your sugar. Okay. If I would have told you to form the guanylate, 7-methyl guanylate, then what would you have formed? You have also added the phosphate group to this sugar that is present here. Okay, so for the 7-methyl guanosine, first you will make the 7-methyl guanine, then you will add the sugar to it, then it will form the 7-methyl guanosine. These kind of examples will be asked in your question papers. Now let's just revise what we had done today. What we had done, we had learned how to draw the structure of a nucleotide and what bonds are involved in the structure of nucleotide. So first of all, we made the structure of nitrogenous base. In the example that I had shown, we had made the adenine. That adenine was joined by the N9 position of your sugar, by the 1 prime position of your sugar, 1 prime of your sugar and 9th position of your adenine. All right, and the ester bond that was formed between the phosphate group and the ribose sugar or the deoxyribose sugar. So two bonds are involved, glycosidic bond and your ester bond is involved, phosphoester bond is involved. And we have also learned this thing that DNA polymerase always work in which fashion? 5 prime to 3 prime. 5 prime to 3 prime. Okay, so whenever it will add a new nucleotide, it will add on the 3 prime of the 3 prime OH of your sugar that is present in your structure. We will learn how the DNA is formed, how the RNA is formed, and what are the salient features of the DNA structure that was actually given by your Watson and Crick, and actually Rosalind Franklin also. So we will learn about that, and we will see the salient features, that is, what is the length of the DNA, what is the length of one pitch, what is the different distance between the two bases, what is the diameter of your DNA. We will learn all of that, and we'll solve certain questions in the next video. So thank you so much. I hope you enjoyed this video. So please like, share and subscribe my channel. Thank you so much. Bye bye.